Okay, I believe you guys in Rochester can hear me while you get seated, and I have a lot of slides, and it's already 1:03, so why don't we get started? So, hi everyone. I'm Eugene Kim, a PhD candidate in MPET program working in Dr. Petruccelli's lab. Today's talk is about the transcriptional inhibition of GGGCC repeat expansion in C9 FTD, frontotemporal dementia, and ALS, amyotrophic lateral sclerosis for the disease prevention and therapeutics. So I'll briefly introduce myself and then give you the background of ALS and FTD, then give you the hypothesis and aims. I'll explain my methods and design, also show you the results, and we will discuss in the end. So the profile. Those of you the first years, I'm Eugene Kim originally from Korea. I finished my doctoral degree in veterinary medicine. I came to United States in 2012 to pursue my research in regenerative medicine in the field of stem cell biology. And I started Mayo Clinic in 2014, which makes me 30 year. By next month, I will be 4th year. And I'm working in neuroscience department in the field of pharmacology and experimental therapeutics. So far last year, I got the grant in collaboration with Veronique in my lab, the NanoSync grant, which is epigenetic targeting of C9orf72. And also last fall, I presented my poster in Mayo Graduate School Fall Symposium. And next month, I will go to AVMA conference as a speaker. So the background. So amyotrophic lateral sclerosis, meaning the malnourishment of the lateral part and hardening of the spinal cord, which is also known as Lou Gehrig's disease. And the clinical manifestation of this disease is unable to move, and the weakness of the motor neurons and loss of the upper motor neuron causes the flaccid, and also the axonal degeneration of lower motor neuron causes the muscle weakness and also the wasting. And another disease that we focus on is frontotemporal dementia, which is the second largest cause of the dementia following the Alzheimer's disease. And as you can see here, the frontal lobe as well as the temporal lobe undergoes the degeneration. And the clinical signs of this disease is the specific deficits in behavior as well as thinking and memory loss. Also, the patients show the language disability as well as the personality changes. So seemingly different two diseases have some common facts such as comorbidities. So the patient initially diagnosed as ALS patient half of them fits the category of the cognitive impairment, and actually 15% of them have meets the criteria for FTD, and vice versa. And also there are familiar cases of FTD and ALS, which is caused by the genetic mutation. And another pathological commonality is the TDP43, which is commonly found in the inclusion of the neurons. And ALS have 97%. The 97% of the inclusions found in ALS comprises of TDP43 aggregation. And this is the genetic causes of FTD and ALS. So if you go to the ALS spectrum, we see that the TDP43 pathology as well as SOD1 and FUS. And if we go further to the FTD spectrum, we have tau mutation and progranulin mutation. And in the middle, there is C9orf72 gene, which has a repeat expansion that I will explain in the next slide. So this scheme shows the pathogenic mechanisms of G4C2 repeat expansion. And as we can see here, the G4C2 repeat expansion locates in the intron region of intron 1 of the chromosome number 9, open reading frame 72. So healthy individuals have 2 to up to 28 repeats, but the patients has 200 up to 2,000 repeats in this region. So one suggested mechanism of the pathogenesis is the enabled to get the C9 or 72 gene itself. So the reduced C9 gene expression causes the haploinsufficiency to cause the disease. And another suggested mechanism is this repeat expansion is somehow transcribed to generate the repeat containing RNAs, which sequesters the RNA binding proteins, or to form a RNA foci, which is a local focal inclusion comprised of this repeat RNA. And more interestingly, there is a RAN translation, which is repeat-associated non-ATG translation that is initiated and caused by this repeat expansion. So based on the different reading frames, it generates 5 different polydipeptide protein repeats. And this also forms an aggregation in cytosol, so it causes the disease. So far, there are lots of evidence that even one of each pathology can contribute the disease, but in a non-mutually exclusive manner. So far, people try to intervene this disease by introducing an antisense oligonucleotide to degrade this repeat expansion, or using the small molecule RNA binders to stabilize this repeat expansion. So I will explain more details about those two interventions. So ASOs. So they are comprised of CCCCGG repeats that complementarily binds to the G4C2 repeat expansion. So this RNA sequesters the RNA binding proteins, and then the RNA binding proteins that includes the RNA related in charge of the RNA processing or splicing event are sequestered by these repeat aggregations, so can result in the malfunction. So antisense oligonucleotides complementarily binds to the repeat expansion and undergoes the degradation pathway through the RNase H. And this is non-structure specific, it is sequence specific. And if you take a look at the RNA of this G4C2, it either forms a hairpin or a G quadruplex structure, which is a parallel. We call it parallel because the topology here goes parallel. So think about the cellular environment. So inside the resting state, the potassium is higher than sodium, so we think it favors the G quadruplex structure, but it goes through the action potentials, and the salt gradient in a local cellular environment enables it switch between the RNA hairpin and the G quadruplex structure. So people try to target the RNA hairpin using a 1A compound, and this is Su et al. 2014 paper. So they used the 1A compound that binds to and stabilize this HRE hairpin to further prevent exert its pathology in cells. And this is C9orf72 positive, meaning that it contains the repeat RNA, whereas negative doesn't. And the poly-GP, one of the dipeptide repeat expansion that I explained before, was detected in the C9 positive cells, but not in the negative cells. So compared to the DMSO treatment, 1A compound significantly reduces the poly-GP level. And this was quantified in 3 different patient cell lines. The foci in the upper panel, as well as the GP level in the lower panel. And another approach is to disrupt this G quadruplex structure using TMPYP porphyrin structure so that it releases the RNA binding protein sequestration. So I came up with an idea that can we then modulate the C9 pathology at the DNA level by targeting the transcription of HRE DNA. So then we have to think about the targets. So the possible target is one secondary structure of the HRE DNA. And also we can manipulate the transcription factors that is specifically plays a role in transcription of the repeat expansion. And thinking about the ligands, we want to find the ligands that can efficiently penetrate the blood-brain barrier to exert its effect. And I came up with this idea because the ASOs that I mentioned before has very limited route of administration to the CSF directly. So in search for the DNA structure, I found this paper in Nature that they show that the possible DNA conformation is G. Now it's the opposite strands goes opposite direction, so we call it anti-parallel G quadruplex structure. And this is in the C. This is a DMS assay which we degrade the DNA and compare the sequence based on the band pattern. So we see that the 1, 2, 3, 4 goes to here, and 6 and 7, 5 and 6 is not degraded, so it forms a loop. So based on this structure and also the circular dichrometry in A confirms us that DNA can form anti-parallel G quadruplex structure. And there also can be anti-parallel or parallel or a mixed conformation. I try to target this one specific structure as it is available in the protein database, which was from the NMR structure that was discovered by Brick et al. And it shows a nice G anti-parallel structure. And for the transcription factors that I mentioned earlier, there is a SPT4 transcription factor that is related to the repeat expression, which is in the context of the Huntington's disease. So Huntington's disease is CAG repeats that generates poly-Q proteins, and for the shorter repeats, as you can see here, even though without the SPT4, UPT4H is the human homolog of SPT4. So SPT4 deletion doesn't necessarily reduce the shorter repeats, but for the longer repeats, it seems SPT4 is required to express the longer repeats. And this is the quantification of this result. So back to the genome biology. So we know that the general transcription factors binds to the promoter and then recruits RNA polymerase II with its C-terminal domain. It's longer here. And once it's bound together, there is a promoter clearance where the G general transcription factors leave the promoter region and the phosphorylation occurs in serine 2 and serine 5 of the C-terminal domain of the polymerase II. And then there is a spontaneous transcripts generated and degraded, and it's sitting there. And once the negative elongation factor and DRB sensitivity inducing factor binds, there is a phosphorylation occurring by the PTEF-B complex, which is a cyclin T plus CDK9, the kinase. So once negative elongation factor is phosphorylated, it's leaving the polymerase II machinery, but the DSIF stays phosphorylated and stays complex with RNA polymerase II to move to the next step, which is elongation phase. So DSIF here is actually comprised of SPID4 and SPID5 that I'm focusing on. So let's take a deeper look at the structure. So the left panel shows that this white is RNA polymerase 2, and the NGN domain and K1L1 is a SPID5 structure. And SPID4, it only binds to the DNA and also to the NGN domain of SPID5 as it's very small protein. And how it maintains the repeat transcription, we think that SPID5 stabilizes the repeat, the unwound repeat DNA to exert its effect. So with this idea, our group last year now deleted the SPID4 in the context of C9orf72, and we saw the nice decrease in the variant 3, which harbors the repeat expansion, mRNA, and also the poly-GP level. And what we found interesting is that not only the GGGGCC, the sense repeat reduces, but also the CCCCGG repeat foci, which is called antisense foci, reduces nicely upon the knockdown of SPT4 or SPT5. But our C. elegans study shows that knocking down the SPT5 was very toxic for the cell, but the SPT4 deletion was doable, or SPT4 was dispensable. So I decided to target this SPT4 with also the...
So the title said that protein selectively regulate C9orf72. So what's the data supporting that selectivity?
So the question in Jacksonville was where is the selectivity part from the data? So
you just showed that protein regulate Huntington expression too. So what do they mean by selectively regulate C9orf72? The title sounds like conflict with what you just presented. So for the Huntington's context, before knockdown was in charge of the long transcription of the CAG repeats. And here we have G4C2 repeat that is harbored in variant 3. And we knock down the SPT4 and we see the reduction, which means that SPT4 is required for the repeat elongation. But you're right. We also saw the SPT5 knockdown also showed the reduction in variant 3, which harbors. Try to depend the authors, but I mean, to me, there is no data showing justify having selectivity there. I mean, whether you agree with the, I guess, authors or don't agree with it, I want to hear your opinion.
At least I can use this data to support my idea that SPT5, the inhibition of SPT4, dimerization with SPT5 can reduce the repeat transcription elongation.
I agree with that. I'm just questioning the title itself.
I think it's maybe I didn't include all the figures that may show this, may support this title.
They lost selectively regulate, right? So I'm just questioning what's their justification. Like, I mean, if they lost SPT4
because in the supplementary figure we show that we tested like 300 gene expressions, right? And we saw that the repeat expansion of C9 was significantly reduced, but not affecting other gene expressions.
So what about Huntington?
Of course, it will reduce. So I think it's selectively, meaning in the C9 patient or C9 patient samples, we see the reduction in repeat expansion transcription.
Because it's selective for mutant. It doesn't affect the wild type, right?
Is that what you want to say?
I think so, yeah. It's
Right,
selective,
right. Selective
to
for
the
the
mutant one
mutant.
with the repeat. And that applies to Huntington, but also C9. But it's
Right.
selective for the mutant. that would be my interpretation.
Yes. So Rochester, here is the summary. So we use the selectively, meaning that it is selective inhibition of transcription in the context of repeat mutations, either Huntington's or the C9 repeats. And now we have to think about the ligands. So there are many ways to penetrate the blood-brain barrier, which is very intact and sealing in the brain for the drug to reach to the brain. We can use the method of passive diffusion, or we can use the facilitated transport, or even the pinocytosis, or using the cellular like a pump. But thinking about the chemical features, we only can predict the the passive diffusion only for the penetration potential using this chemical features-based prediction. So I was lucky to find this company, which is called Cambridge. They have this CNS selection pool, which has all the physical chemical properties that can penetrate the blood-brain barrier. So they have lower molecular weight than the Lipinski's 5 rule of which maybe MPS students are familiar with. So it applies more strict rule for the drug discovery. So it has a hydrogen bond and donor and acceptor number, limited number of 8 and 5, and total polar surface area of 120, which is very strict, and also has a limited number of rotable bonds. And they also did all the Keiko cell penetration assay, which shows that it has potential to go through the gut-blood barrier to once it's administered orally. So I decided to use these CNS compounds. So my hypothesis is that we can inhibit the transcription of G4C2 repeat expansion by using the blood-brain barrier penetrating small molecules that directly bind to the DNA G quadruplex structure. And my second aim is to inhibit the SP4 dimerized with SPID5 to decrease the C9 pathology, such as variant mRNA transcript level that contains the repeat, or RNA foci, or dipeptide repeats, therefore providing a neuroprotection effect against G4C2 associated toxicities in C9. So the specific aim one is to utilize a bioinformatical approach to select the best binders of G4C2 DNA. And I will use the in silico drug screening method using the Cambridge CNS library. And secondly, I will examine and validate the biological activity of the small molecule binders in HEC293 T cells to determine the dose and the toxicity. And lastly, I validate the hits in eye neurons and lymphoblastoid cell lines to measure the drug effect in C9 pathology. And second part of my aim is targeting the SPT4, and we will use the peptide design approach to target the dimerization by designing against NGN domain of SP4 and 5. And we'll create the system that can test this peptide affecting the dimerization by generating a split luciferase assay system to closely monitor the dimerization. And lastly, we will validate the hits in physiologically relevant eye neurons, induced neurons, or lymphoblastoid cell lines. So let me explain how the workflow. So I will do the in silico screening and identify the binders in vitro and examine the activity of the binders in HEC cells, also validate the leads in the eye neurons, and finally test the compounds in mouse model. And in silico, we will use the data from the eye neuron to feed back to the in silico drug design to find the better compounds. So the methods. For the in silico methods, I used the drug screening using a autorafina program. So I target the having the 2 and 2D, which is a G quadruplex structure that I showed before, and with the ligands of the Cambridge FDA approved as well as designed, I dock it with the AutoDocVina program to find the affinity. And I do the similarity search of the top binders and have second run to screen and then choose the ones that has the highest affinity and cluster them based on the similar physicochemical properties and then end. So this is an example of running a tetracycline, which is one of the FDA approved drugs docked in 2 different macromolecules, which is G quadruplex and a hairpin DNA. And it has a 10 confirmation of possible drug confirmations, and I picked the highest affinity score with the most likely confirmation, which was -7 and negative 7.9 and 7.5. And then in order to generate the preclinical model that I can test my compounds, I took C9 negative as well as C9 positive patients' blood samples and isolate the lymphocytes and introduce the Epstein-Barr virus to generate lymphoblastoid cell lines, which is floating cells and easy to culture. And I treat DMSO as a control, and then do the RNA foci, poly-GP, as well as the qPCR to check the variant RNA level. And another cell model is the more physiologically relevant cell model that's from the patient's skin cells to differentiate into eye neurons to test the compounds. So I test the compounds and then do the same assay to measure the C9 pathology. And we use the method from this paper by Zhu et al. So knocking down the polypyrimidine track binding proteins can enhance the neuronal expression of the neuronal morphology of the fibroblasts. And by the day 5, they show the nice external or dendrites and has all the neuronal markers such as GLUT1, MEP2, or synapsin1, which is an indication of the synapse. And this is my differentiation method using the same protocol and plus the small molecule inhibitors. So it shows nice neuronal morphology by day 10 and maintains the morphology until day 26. And lastly, the qPCR method for detecting the multiple variants. So variant 1 and variant 3 contains the repeat expansion, whereas variant 2 doesn't. And the detection primers were designed based on this. So we have variant 1 detection from exon 1 to exon 2, and variant 2 we detect from exon 1b to exon 2. And lastly, variant 3, we have this primer. So since variant 1 is shorter than variant 3, the variant 3 can be selectively detected by using this portion of the RNA. And I will explain 2 more technical methods to detect the C9 pathology. So we have FISH, which is fluorescent in situ hybridization. So I set the cells in poly-D-lysine coated plate, and I fix the cells, and using the complementary dye that has a fluorescent probe in it, I do the hybridization and wash and image. We can get the nice image that shows this fluorescence. And MSD assay is mesoscale discovery assay, which is highly sensitive method that is suitable to detect the very little amount of dipeptide proteins. So it's similar to the sandwich ELISA method, and it has a capturing antibody, and we put the analytes, and it the sulfotag detection antibody is applied. So we can detect the electrical change as well as the chemical change and also the light change. So it's highly sensitive. So the result. So I docked the FDA compounds, which is 1400, and got the top 10 here. So digitoxin, which is known very promiscuous drug that can bind potentially to like 300 targets, and it is not surprising it came on top. And 3 compounds are tyrosine kinase inhibitors and also some the drugs that is used for bipolar disease or a cardiac arrhythmia. So when I group them together, the RTKs cluster together here, and this time I included 1A, which is RNA hairpin binder, clustered together with thelithromycin, whose structure is depicted here. So based on the physical-chemical properties of FDA, it will be almost impossible to repurpose this drug because of the high molecular weight and the polar characteristics. And then next I moved to the drug screening of CNS compounds, and I screened 58,000 compounds, and those are the top 10. And the affinity range from 12 to 13.4, which is pretty high, and the structure are depicted here. So 1 and 2 shares the common structural similarity, and 7 and 9 shares also the similarity. And the list goes on. So I will show you how it fits into the G4C2 repeat expansion. And in courtesy of Tom Caulfield, I visualized the binding of the number 6 compound, and if you closely look at it, the hydrogen bond from the phosphate group of the DNA nicely forms a hydrogen bond to the drug. And the drug, since it was folded like has a kink structure like this, we have nice insertion deeply to the groove of the G4C2. Thank you. Yes, with the sulfate group, phosphate group to the internal direction. So using these 10 compounds, I tried to get the toxicity level by doing the lactate dehydrogenase assay at 10 micromolar, and it showed the least amount of toxicity. So I moved on to the RNA foci assay, the fish, and among 10, 9 is showed here, and 1 compound was toxic at this concentration. But note that the top 3 compound here was significantly reducing the RNA foci in lymphoblastoid cell lines. And this is a poly-GP level that was detected by the MSD assay. And here it shows that the 2 compounds here, 224 and 945, shows a nice reduction in poly-GP. So I decided to use the 224 compound for further assay in induced neuron. And I didn't want to give up on this 1 compound that shows toxicity, so I used multiple doses from 0.5 to 1.5 to detect the mRNA level. And it significantly reduced the mRNA variant 3 level, which harbors the repeat expansion, at 1.5 micromolar. So I decided to use this 1.5 micromolar of this 200 compound for the future study. And this is another RNA foci assay as well as poly-GP level of that compound, and it showed nice reduction in a dose-dependent manner. And before I move on to the eye neuron, I did a time course cytotoxicity assay using LDH. And the white shows the 200 at 1.5, and the gray shows the 224 at 10 micromolar. And we can see that the 200, as the time goes by, it gets more toxic to the cell, whereas the 224 stays almost the same, or even lower than the DMSO treatment. And this is image of fish, which shows DMSO treated O203, which is C9 negative, and the C9 positive cells, O220, was treated with DMSO and 2 other compounds. And we can see the nice foci in the red dots here. And I quantified this by doing...
Fusion?
Yes?
Before you going further, in slide 51,
This? No.
probably 52, I think. Yeah, that one, 52.
Here.
Sorry, one previous one.
52. Oh, yeah. No. Here.
Yes.
Yes.
No, can you go one more back?
Here?
Yes. So here, didn't you say C9 variant 2 doesn't have the repeat region? So probably I'm not understanding the graph. So if your treatment reduces the B2 level as well as in variant 3, what does it mean?
So unlike the RNA binding compounds, we are targeting at the DNA level, right? So if the compounds bind to the multiple, like hundreds of the repeat expansions, then the RNA polymerase 2 cannot go through the intron 1. So that's my purpose of preventing the V3. And V2 can be generated by the splicing event or even using that repeat expansion as a promoter. Let me show the So let's think about that variant 2. So variant 2, the promoter region is within the repeat expansion part. So if we use the compounds to bind to the repeat expansion, it may affect the promoter activity for the variant 2. So I think the variant 2 level also may be affected by the compounds.
Yes. So although you show the overall level of C9 transcript level hasn't been decreased, so if you take non-discriminatively affect all the transcripts, you showed that one of the pathological mechanisms is haploinsufficiency.
Yes.
So that can be still covered by the wild type allele. So even though you affect all 3 transcript types, it should be fine therapeutically. Yeah.
So so far in the field, the gain of toxicity caused by the repeat expansion is more major opinion than the haploinsufficiency theory. So I cannot say that the compound binding to the repeat DNA reduces doesn't reduce, I cannot say that doesn't reduces the variant 2 level, but if you see the overall C9 transcript level, it's not significantly reduces. So I think we still can compensate that C9 gene haploinsufficiency issue.
Sure, thank you.
Can I ask you a question? Have you tried the compounds on a control cell line that doesn't have the repeat?
Yes, it's this is the cell doesn't have a repeat.
Well, of course, I know both sides, but I mean the levels of the transcripts.
So the question is, have I tested the compounds on a non-repeat containing cells? I only tested on hex cells to measure the toxicity, but other than that, didn't pursue it with on the cells without the repeat. And what's your...
Yeah, I mean, because if you want to see if it's really specific for the repeat, you could compare an ALS and a control cell line, for example, lymphoblastoid cells or eye neurons or whatever. And if it's really specific for the structure, for the G-quadruplex structure and for the repeat, it should not affect the transcription in the wild type. The not mutated, the repeat.
So what's your readout for that specificity?
Well,
you want to read the
you
older
look
gene?
at the different transcript levels, right?
Yes.
And you would predict that the transcript levels are not affected in a control cell and it does not have the repeat, right?
Yes. We have a great comment here. So if we don't have a repeat, then the drug shouldn't affect any of the level of C9 mRNA level.
Right.
Because, yes. That's a good point. So the quantified eye neurons has the significantly reduced foci in both treatments using two different drugs, and also the poly-GP level reduce after 13 days of treatment. So summary, if we have the repeat expansion opened in certain cellular events such as transcription event, we can target that G-quadruplex structure using the small molecule inhibitor, and we can further reduce the variant containing RNA level as well as reduce the dipeptide repeat expansion by reducing reducing the level of the variant 3 mRNA. And the part two, I will explain the assay development and one peptide result that I have now. So let's see. The round one is SPID4, and it's tagged with the half construct of luciferase gene. And when it's bound with SPID5, which is larger here, with luciferase half construct, it forms a dimer. And when it forms a dimer, the luciferase retrieves its activity. So when we add the substrate, which is covalent tyrosine, it gives a luminescent signal. So in the presence of synthetic peptide that I will discuss later, if we have that is bound to the binding site at the SPID4, we cannot form a dimer, so we have no signal. So how do I make the constructs? So in two-dimensional, it's simple to think that maybe let's say here, up here is N terminus and here is C terminus. So C terminus tagging may be efficient, maybe enough, but we can think three-dimensionally. It can be N-terminal tagging both, or C-terminal and N-terminal tagging on the SPID4/5 may give the highest luciferase activity. So I tried to make 4 different clones using this SPID4/5 / luciferase 1,2. And this is what I'm saying. So I did a Western blot after transfecting these 4 different constructs, which is luciferase in N-terminal or C-terminal, and vice versa for the SPID4/5. And leucine zipper was used instead of the 4-5 because zippers closely form a dimer, or zippers from the dimer that can be used as a drug treatment control, as it gives the signal all the time. And immunocytochemistry results shows that the SP4 and 5 overexpression is in the nucleus, and which mimics the endogenous SP4-5. And using these constructs, I decided to do the Gaussian-luciferase assay, which is pretty simple. So I transfect the cells and treat, or without treat, I remove the media and add the luciferase substrate, and then read the signal at 480 nanometer. And the result is here. So when we transfect the single constructs, it doesn't have a signal, but when we transfected the SPID5 LUK2, we have some signals here. Either way, the SPID4 tagging, SPID5, the luciferase tagging in N-terminus gives the high signal. And from now on, I used SPID4 LUK1 and SPID5 LUK2 for a drug treatment assay. And this is co-IP data showing that the endogenous SPID4 is 716 and overexpression SPID4 is 26. And near 134, the endogenous SPID5 is 134 and 136 is one overexpressed. 150 is overexpressed SPID5. So input doesn't have a significant difference in the level of SPID5 here. But when we pull down with SPID4, which binds to the SPID5, we see that the non-overexpressed SPID4 doesn't pull down the SPID5. And this is another control for the antibody, that when we pull down with SPID4, we pull down the SPID5 together. But when we pull down with just regular rapid IgG, we do not pull down the SPID5. So the co-IP data is consistent with luciferase assay, which shows that the luciferase signal is reflected of heterodimer formation. And this is a sequence map of SPID4 and SPID5. So SPID4 is pretty simple structure. So it has an NGN domain that is a binding domain with SPID5, and also zinc finger domain that is binding to the DNA. And the alpha 1 of NGN domain of SPID5 is interaction domain with SPID4. So we decided to remove this beta 4 strand to see whether it can abolish the dimerization. And this is a structural prediction that showing that the deletion of this beta 4 doesn't affect the overall conformation of these proteins. And the immunocytochemistry shows that the nice overexpression in the nucleus of the deletion mutation, as well as I made a point mutation as a control. And this is a co-IP data again, showing that the deletion mutation failed to pull down the SPID5 overexpression, which is suggesting that it is critical domain. And this is when I use anti-Gaussian luciferase antibody, showing that the third lane of the right panel here, we do not see the SPID5 pull down together with SPID4. So I went ahead to do the luciferase assay, and the white bar shows the wild type, and the black bar is the point mutation, which doesn't help reducing the interaction. But when we see the deletion mutation of SPID4 together with SPID5, abolish the luminescence signal. So now we know that the serine 56 through serine 69 is very critical domain for the binding. And lastly, to test the specificity of this assay, I went ahead to use the alpha-synuclein tagged by luciferase 1. And on the right panel, it shows very little luminescence signal, which suggesting that the binding of SPID45 luminescence signal of SPID45 binding is specific for the dimerization.
But the anti-blue, are they the same metal?
Yes. This is showing the anti-look, showing the overexpression of each construct with alpha-synuclein or with SPID5 and also the leucine zippers. And using this system, since I've established this system, I try to use some of the FDA drugs and also the designed peptidomimetic drugs. And here I will explain you majorly the peptidomimetic approach. So peptidomimetic is mimicking the protein that is binding to each of the protein, and I will use the sequence of the protein region together with arginine-rich sequence to enhance the cellular membrane penetration. And this is the sequence that I choose to try from the SPID4 and from the SPID5 at their interaction surface. And this is a list of peptides that I want to try, and I will show you the result from the peptide 1 and 2 treatment. So upon the treatment of peptide 1, it shows a nice dose-dependent reduction of the signal in a specific manner, as it didn't reduce the signal from the leucine zipper control. So in summary, the 7731-30200 compound significantly reduces the C9 pathology in physiologically relevant model in eye neuron and LCLs. Also, the SPID4 tag, C-terminal tagging of SPID4 and C-terminal tagging of SPID5 pair gives the highest luminescence signal, which can be further used for a drug screening purpose. And specificity of this assay was verified using a deletion construct as well as using alpha-synuclein constructs. And peptide arginine plus the chunk sequence from the SPID4 was significantly reducing the luminescence signal at 20 micromolar in hexels in a dose-dependent manner. So, of course, I will do more validation using different sets of peptides, and in the future, I will do the mouse study to get the pharmacokinetics and pharmacodynamics of the repeat binders and do the behavioral study as well as the histopathological study, including the RNA foci, OGP, and a behavioral study. With this, I'd like to thank my PI and Dr. Zhang, as well as my lab members enlisted here. And also thank for the committee and MGS program. Thank you. So we have 5 minutes, so we will take the question from Rochester first.
Not related to the material. So within 2-year time span, you screened close to 100,000 of compounds while doing hardcore molecular biology, making multiple types of mutants. How many hours do you work per day? As a fellow graduate student, I feel like I should leave the field.
No, Han, it's for the screening part, I do the in silico screening. So I tested only 10 compounds, and I just work regular hours like other students.
Wow.
So you guys wonder why peptide 1 worked, but not the peptide 2? Yeah, because I used the same sequence from NGN domain from the SPID4 and also the SPID5, but the SPID4 chunk that is bound to SPIP5 abolish the dimerization, but the other way around it was not, suggesting that SPIP4 has like much lesser surface area, so the only way that it can interact to the SPIP5 is limited surface area of the NGM5, the SPIP5, I guess. Yes, Pam.
As an additional control, did you look at SPIT4 GLUC1 and SPIT4 GLUC2 together, and SPIT5 GLUC1, SPIT5 GLUC2 together?
Dr. Pam said, as an additional control, did you try the SPIP4 that is tagged by LUC1, LUC2, or SPIP5 tagged with LUC1, SPIP5 tagged with LUC2? I haven't, but
Do they form homodimers?
In my opinion, it's not gonna form a dimer because even NGN domain has similar like beta sheets, like 4 beta sheet strands binding together. It should be the same side NGN domain wouldn't dimerize together. That's my thought, but I definitely can try it. That's easier experiment.
It's just a relevant control, you
Yes.
know, with instead of the synuclein with the SPIT4.
Right. That can be a relevant control than the synuclein control.
So I really like the attempt to try to interfere interaction between step 1 and step 5. Before we jump on drug screening, is there any, say, some genetics data suggesting actually that's a good drug?
So is there any good genetic study suggesting that SPIP4 is a good drug target? That's a good question.
Interaction between 4 and 5. This one is
4 and 5.
Here the interaction between step 4 and 5, right? But
Right.
is there any good rationale why it's a target? I mean, you need strong rationale to convince yourself to invest so much time doing drug screening, right? Because screening itself is high risk already. But you need to be sure that you think that is.
So Dr. Kim here commented that SPIP4 is really good candidate. Do you have any supporting data since it's labor intensive to do the drug screening? We have the knockdown data, but there is no other group tested or tried to interfere the dimerization.
It's interesting, but I'm just curious. Justify that.
Right. I think the SPIP4 knockdown in Huntington's and also the last year's our C9 context can be a supportive data for me because deletion of the SPIP4, like 100% version of the interaction, is giving a more than 50 or 70% reduction in RNA foci and GP level. So it's highly encouraging that if we successfully can interfere the dimerization, and it seems like the only targeting pocket for the SPIP4 is the NGN domain, then by doing the computational analysis and using this system, we hopefully can find a good compound for
Is there
targeting.
other normal proteins, I guess normal genes that step 4 regulate, other than those diseases?
So the question is there other normal gene that is
Regulated by step?
regulated by SPP4?
I assume so, right? Doesn't make sense to have a protein just
Have
cause
a-
disease-causing mutation.
Yeah, I believe there may be the repeat containing genes.
There are many, right?
There are a bunch, and there is telomere sequence that needs to be, yes.
I guess another way of thinking of what Jung is asking is what if SPIT4 has functions that don't require heterodimers with SPIT5? So knockdown of SPIT4 can reduce RNA foci, but what if it's got some other partner or can operate as a monomer or something?
What are the binding partners of SPIP4 that can exert its activity not through the SPIP5? I don't think so because there is no interaction motif other than NGN, and if we abolish its docking with SPIP5, then even though it has a DNA binding zinc finger, it solely with itself cannot show its activity, I assume. So another study that without my thesis work, I'm trying to find a modification site of SPIP4 that may be another target for its activity, not through the SPIP5 interaction.
Is it clear whether these G-quadruplex structures are formed in vivo in the context of a live cell? Has anybody used any probes to see, for example, some small molecules that specifically bind G-quadruplex structure or the BG4 antibody to see if these structures are really present in cells? And not
That's very valid point. So is the G4C2 antiparallel structure exist in the cell? So not in the context of G4C2, but I have seen the telomere binding RNA, the fluorescent probe that can detect the RNA G quadruplex structure of telomere in cells. So I was thinking we can use that to detect the same with using that approach.
I think that would be really important because most of the studies that I'm aware of have been done in vitro, and I don't think it's really clear whether.
Yes. So yes, I think most of the times the DNA exists in a duplex, right? Not the helix structure with its complementary binding. But in the event of transcription or when it needs to open, like I showed in the scheme, it definitely can form its own G quadruplex or the anti or mixed population, which was already shown by other groups. And also the CCCCGG, the complementary strand can form its own I motif or other secondary structure that is favored more than the helix structure when it's opened. So I guess there is a structure that when it opens, the structure fluctuates, or shifts between antiparallel or parallel, but there is evidence that the G quadruplex is the most favorable structure for the DNA structure in thermodynamically. So if you spontaneously unwind the DNA, then G quadruplex antiparallel forms first, and as the time goes by, it transitions to the parallel and a mixed G quadruplex structure. Do you have any more questions?
Nope.
Thank you for your precious time to attend my talk.